Hello, everyone, and welcome. Thank you for joining us today for the third webinar in our current series titled The New World Screwworm, Past Lessons, Present Programs, and Future Frontiers. My name is Justin Overcash, and I'm with the Foundation for the National Institutes of Health, where I work as part of the Gene Convene Global Collaborative. Gene Convene's mission is to support and advance best practices and informed decision making for the development and governance of genetic and biological control technologies to improve public health and animal health. We do this by convening diverse experts, identifying key scientific and policy questions, and supporting transparent information sharing and capacity building across sectors. This webinar series focuses on the New World Screwworm, a pest that has historically caused devastating impacts on livestock, wildlife, and human health, and whose successful control stands as one of the most significant achievements in area-wide pest management. At the same time, recent detections and northward spread underscore that it is not just a historical story, but a contemporary and forward-looking challenge. Across this series, we will examine past lessons from eradication efforts and international coordination, present programs, including surveillance, preparedness, and containment, and future frontiers, including new scientific tools, technologies, and policy approaches that may strengthen long-term control and prevention. Before we get started, I'll cover a few housekeeping reminders. If you experience any technical issues during the webinar, please email events@fnih.org. Your video and audio will be off during the session. Today's presentation will run approximately 45 to 60 minutes. Please feel free to submit questions at any time using the Q&A function. We will address questions during the discussion period at the end of the presentation as time allows. This webinar is being recorded, and recordings will be posted to the Gene Convene Virtual Institute as well as the Gene Convene Virtual Institute YouTube channel following the event. Today, we are pleased to be joined by Dr. Paul Hickner. Dr. Hickner is a research ecologist with the U.S. Department of Agriculture's Agricultural Research Service, or also known as ARS, based in Kerrville, Texas. Dr. Hickner's research program is focused on improving the management and control of the New World Screwworm. His graduate work at the University of Notre Dame examined the genetic basis of key adaptive traits in Culex pipiens, mosquitoes, including reproductive diapause, autogeny, and blood-feeding behavior. Work that grounded his expertise at the intersection of genetics, physiology, and vector biology. During his postdoctoral research, Dr. Hickner investigated the genetics basis of olfaction in mosquitoes, fruit flies, and sandflies. Since joining ARS in 2020, he has focused on developing novel attractants for New World Screwworm and advancing our understanding of their chemical communication and mating behaviors. His current research integrates genomics, physiology, and chemical ecology to develop sex-specific attractants that could enhance surveillance and control strategies for this economically significant pest. Before we begin, a quick reminder that I'll be moderating the discussion following the presentation. Please feel free to post your questions in the Q&A at any point during the talk. As mentioned earlier, the webinar is being recorded and will be made available following the event. With that, I'll turn it over to our speaker. Dr. Hickner, whenever you're ready, please go ahead and share your slides.
Can you see my slides okay?
Yes, we can see your slides.
Okay. Good morning, good afternoon. I guess it kind of depends where you are. It's morning here. As Justin mentioned, I am a research ecologist at ARS, USDA ARS in Kerrville, Texas. This is where the Screwworm Research Group works out of if we're not in Panama. So we do, some of us go to Panama and we actually do rotations through there. And my main focus is the development of novel attractants. I also do other things. I am going to spend time talking about some of our genomic work first. We just had a paper released earlier this week describing the new genome for New World Screwworm. So I'm going to talk a little bit about genomes, some of the projects we're doing there, and then how we're leveraging that data to develop novel attractants. And I am sure you are heard if you attended the previous webinars. You probably have a pretty good understanding of screwworm, but in case you did not, I'm going to just go over some basics. New World screwworm is an obligate parasite. Um, Affects many animals, but of a livestock concern primarily, but also a human health concern. Um, it was eradicated using the sterile insect technique, eradicated from North and Central America during the end of the 20th century. Down in the bottom of this bottom left, you can see this is this is the Copeg facility that is currently producing um, 100 million flies a week, approximately. And you can see a very large plant. And it's a factory of rearing flies. So basically we rear flies, they get sterilized, and then we release them to mate with the wild flies in nature. It's primarily driven by it is driven by the sterile males. So you will hear us talk about this in a little bit different methods to do some trapping here in a few minutes. So Cochliomyia, the genus, there's 4 described species. We hear about New World screwworm, which is Cochliomyia hominivorax, and it pre-eradication distribution was basically not temperate areas. You know, if they went into temperate areas, it would be just during summer months. But it ranges, you know, through a large part of the Americas. A common species, even more common than the New World screwworm, is Cochliomyia macellaria. We have these, they are very common throughout even temperate areas of the Americas. They are typical, what we'll call a typical blowfly. They rarely cause any kind of myiasis. They typically feed on carrion. So a lot of roadkill. When we go to collect them, we often collect them from roadkill. But they're very closely related to New World screwworm. There are a couple other minor species. We'll call them minor just because they're not as broadly distributed as the primary or New World screwworm and secondary screwworm, Cochliomyia macellaria, and that is Cochliomyia minima, which is the mostly Caribbean in the purple circle. So it broadly distributed across the Caribbean. And then we have Cochliomyia aldrichi, which is a little narrower distribution. One of our workers here, researchers here, did a lot of this work in the Caribbean, and she found it only on one island when she did her study. And parasitism in blowflies is not uncommon. Lucilia caprina is a major pest, especially in Australia, where it kind of comes in 2 forms. The Australian form that parasitizes sheep is pretty much, I know we talk about it as being a facultative parasite, but you know, they rarely find that form in dead animals. So it just comes in 2 forms. One is almost or practically an obligate parasite, while the other is we see here in the United States, and it's just kind of a typical blowfly. We have some Ballardia species. That's why this earthworm is here. They are believed to be parasites or parasitoids of earthworms. Of course, we have New World screwworm, which infests a lot of livestock, but also wild animals, rarely humans. And then we have some obligate parasites that are blood feeders, actually, in a larval form. So several forms of parasitism occur in the blowflies or the Calliphoridae. So I started getting involved with New World Screwworm back in around 2018, 2019, when the first genome was sequenced. Um, I did the chemosensory gene family annotations for that genome, that first genome, from basically the Scott lab, Max Scott lab, led that. So I'm going to talk a little bit more about chemosensory gene families down the road, but quite variable. They're very interesting in that they kind of reflect oftentimes the habitat, their niche. Are they generalist? Are they very specific? Um, are they very specialized? So as you can see, they vary in some of them have a lot of odorant receptors, some have very few. So it's they really are dynamic, the way they expand and contract. That genome, that first version of the genome was later scaffolded using Hi-C. And we just published, just came out this year or this week, a new screwworm genome. We used a little bit different approach this time, but the first time around, the technology didn't allow us to um, really sequence, utilize an individual fly. And we had to use multiple flies. So we had problems with heterozygosity. Now technology has allowed us to just sequence a single fly. And in this case, what we were able to do was we took a Yavisa male. So this was a line that was colonized from Panama in the Darien region, probably around 2021, 2022. And that's how we start new strains. Basically, we get them from myiasis cases often. But we took a Yavisa male and we took the current production strain at the time was J06. It was basically a Jamaican strain, colonized in 2006. So we did a crossing scheme where we took a male from Yavisa, a female from J06, and then we sequenced the F1 offspring. And what that does for us is it allows us to actually sequence the individual haplotypes. So what it produced was one haplotype for Yavisa, the paternal, and a maternal haplotype. And what we did was we took the paternal haplotype and moved the X chromosome and mitochondria over there, and we now have a reference assembly and a haplotype 2. This worked really well for us. The first genome, and I do have some data here on the first genome. When I mentioned the heterozygosity and some of the problems associated with that and doing multiple individuals, what happens in many cases is they don't get collapsed properly. And you'll get a genome that's a little bit too large. And that's what happened the first time around. Or the genome from that early, I call it the 2023 genome. That's because that was the genome after it was the Hi-C scaffolding manuscript was published in 2023. But it was 534 million bases. Flow cytometry suggests that it should have been smaller. So when we did our newest genome assembly, we did get closer to the estimates for based on flow cytometry. So we got about 455 to 465 million bases. So we believe we have resolved a lot of the uncollapsed haplotype issues that we had in the first assembly. We were also able to get the sex chromosomes a little bit better assembled. Right now, they stand the X chromosome in hominivorax stands around 7 million base pairs. And the Y chromosome, which is really left us scratching our heads a little bit. It seems a little small, but we keep coming up with the same answer. We've done a couple recent sequencing, MorePack bio sequencing, and it keeps coming up around that 600 or 0.65 million base pair size. So we believe we have, if it's not complete in that size, we have at least the most important part, and I believe we have what we think are male determining genes there. But we are still in the process of confirming those. So we also used the new NCBI's, they call it EGAP-X. I forget what that acronym stands for, but they were doing a really good job with annotating certain genomes. So they created a pipeline. So they rolled that out to the public, and we are now able to use it because I think they were getting pretty much overwhelmed with requests to annotate genomes. So this is something that we can do on our own using the NCBI pipeline. So we use that here, and we got a gene number that we feel is we're pretty confident in. The total number of genes in our reference in our haplotype 2 were a little over 13,000. 4,000 plus have variants, splice variants that were described. About 800 to 900-ish non-coding genes, and around 12,500 protein coding genes. These are complete. Of course, we run the BUSCO evaluation of these, and they came out really good. What it looks like the genome is highly complete, as is the annotations. So we also did the secondary screwworm fly. So this is, I'll show some of this data here in a few minutes, but we really want to find things that are different between the New World screwworm and the secondary screwworm to kind of help with our specificity of trapping. So we did the Macellaria genome in the same way, the same strategy. We call this trio binning. This was a cross between an NC State strain and a Kerrville strain. And interestingly, and this is not surprising at all, this is what we would have predicted, that it has more genes. We found a total of 14,500 genes and around 13,400 protein coding genes, which is less than the New World screwworm. And this is predicted because obligate parasites, it's a form of trophic specialization, so they don't do as much as the secondary screwworm fly, which gets on carrion. It can live at various different temperatures. New World screwworm only lives in nature, only lives in living animals, very specific to temperature. So, it evolved in living animals. It's very narrow range of temperatures, at least in the larval stage. So it's a more compact genome. It doesn't do nearly as many things as a generalist like the Cochliomyia macellaria. A larger genome as well. And we're also doing we now have all of the Cochliomyia sequenced. Here's a picture. I think these are really interesting looking. This is Cochliomyia minima from Puerto Rico. Very similar in looks. It has these stripes here on the thorax. But what's very interesting that's different with minima is it has this very shiny, coppery looking tip of the abdomen. So we sequenced Cochliomyia minima, we sequenced Cochliomyia aldrichi. We did those initially through samples that were collected back in 2012, and they were stored in ethanol, which we didn't think we would be able to get long-read pack biosequencing with those samples, but we did because they were taken care of. The alcohol was changed, it was kept in a freezer, and we were able to get full genomes. Now, we weren't able to scaffold them because you need fresh tissue for that, but we have very nice, I think, genomes for Cochliomyia minima and aldrichi. So we also kind of asked the question, what happened to the genome during this evolution? Hominivorax, we think the common ancestor is probably a typical blowfly to the hominivorax here. So you can see this is Compsomyops callipes. Looks very similar to the Cochliomyia, very closely related to the Cochliomyia. So these, we got samples from California. They are here in Texas, but they're not real abundant, but they are really abundant in California. But they're a typical blowfly, pretty large blowfly. But we're going to use those. That's now our outgroup to study the Cochliomyia. So we want to look at what's changed over this evolutionary time in the obligate parasites genome. And we have estimated that. I did not put that in my talk. I apologize. But we think this all happened within the last 10 million years. And with these minima and aldrichi being just a couple of million years ago. So we do have that coming up. We will do a quite broad analysis of those genomes to look at birth and death evolution, how many genes were lost or gained during the evolution of parasitism. We'll also look at signs of signatures of positive selection. And like I said, we've already done the divergence time estimation. So we hope to have that analysis finished up this year. But now I'm going to kind of talk about how we use these data to develop novel attractants. I know that might sound unconventional to a chemical ecologist, but there is some a method to our madness here, so you'll see. So traps have been used for New World screwworm for 100 years. And this is a picture from the 1930s, mid-1930s. And this was done in Texas. So this is a trap capture from what they used back then is what we call a bishop trap. And they would just use rotten meat baits to collect all the blowflies they could. And they would get a lot of them. So they're I like to think of them as very olfactory driven. They can be lured in. Interestingly, this pile, which is who knows how many millions of flies are in here, back during this time, they didn't understand completely which species was causing the myiasis. They thought perhaps it was many species. And shortly after this picture was taken, they determined that it was only one species that was really causing all the myiasis. And and it's hard to say now, but based on what we have done with our trapping efforts over the last few years, we would probably expect less than 1% of these are truly New World screwworm. Just because New World screwworm is not typically very abundant in nature. And that's one of the reasons why I think that SIT works so well with them. When we trap go set traps in the tropics, in Nicaragua or Panama, we'll catch a lot of macellaria. I think the last time we did a trapping, a field trip, we caught 3,500 macellaria and 31 New World screwworm. So just in nature, these other blowflies are just more abundant. So very few of these are going to be New World screwworm. But traps were used for surveillance, currently used for surveillance and to assess dispersal of the sterile released flies. One example was, sometimes it's hard to get a plane, get flies dropped exactly where you want them due to prevailing winds or how close the plane can get down. So they can go in and assess if those flies are being disseminated where they're supposed to be, using traps. And of course, back in the 30s, this was what they were using for control. Only later, a couple of decades later, did they really use the sterile insect technique. And then since then, that has been the most effective approach for controlling screwworm. We do believe that traps still have an important part of our control program, and I'm going to talk a little bit about some attract and kill strategies that we are working on. So the early traps used rotten meat baits. It was in the Bishop trap, is what they called it. We still use rotten meat baits. So this is we use rotten bovine beef liver right now, mostly for catching live flies. If we want to collect flies, in our swarm lure traps, I'll show you in a minute. It's an adhesive trap, so flies get pretty much ruined. They're dead when we get to them, and that adhesive really does a job on them. If we want flies that are in better condition, then we'll just go and net flies. So that's what this is John Welsh, someone that was with the program, has been with the program for decades, and they just put out aged beef liver, and they will capture the flies with a net as they come in. And what's interesting is that we catch different things with liver than we do with our other baits, our other attractants. Liver is catching almost all females. We are not catching males, very, very few males with liver. And we believe that's because, like a mosquito or like many flies, the females need to take a protein meal to help develop the eggs. So they're coming in and they're taking protein meal. The males are not going to be taking those protein meals. So we think this demonstrates the ability to develop a chemical attractant that can be female specific. And that's one of our objectives that we're shooting for. Our current attractant, we call it swarm lure. First developed in the 1970s. It's gone through several iterations since then. The most recent was Swarm Lure 5. That change was made just to, we couldn't ship dimethyl disulfide via air anymore. So due to its toxicity. So we did some of our earlier work showed that they do respond to dimethyl trisulfide, which we could ship. So that was the main revision to swarm lure at that time. But this chemical lure really allowed the program to innovate in trap design. Up here in the center, this is the trap they were using up until last year. So this is I believe it's like a fiberglass sheet. And it's a black sheet covered with, if you've ever used some of these insect adhesives, that glue. So they paint that on there. And this here is a wick, and that's where they would apply the swarm lure. There's two of them. There's one on each side, right in the middle. This thing will spin with the wind a little bit, and it's about 1.5 meters off the ground. Here's what we call the vertical sticky trap, which is this first trap, and our newest trap, the cylinder sticky trap. This is what they are currently using. And I'm going to see if this will play. It will show you kind of one of the reasons we think this cylinder sticky trap is better. You'll notice that vertical sticky trap is really moving. It's whipping a lot. The cylinder trap is not. So we feel like the flies are better able to land on that cylinder trap. It's just a better visual target, and it's just not moving as much. So that is the current trap that we are using right now, is that cylinder trap. Some of our recent studies have shown that, Swarm lure 4 and Swarm lure 5 are about the same in the proportion of males and females collected. That doesn't do a great job on just females. It's about 70% females, 30% males. We would really like to decrease the number of males captured if we are going to employ an attract and kill system. The idea being, like I mentioned before, the males are what's doing the work, the sterile males that we release. If we can get those sterile males to the wild females first, then those wild females will not ever have lay fertile eggs. So, but we produce males and females in roughly equal proportion. So removing any of these females from the population would only be a positive, right? Not only could we be removing gravid females that have already mated and are getting ready to lay fertile eggs, we could also remove some of the sterile females, and we believe that would help the efficiency of the system, where we can get the fewer steriles that the sterile males are competing with or competing for, the better. So the first aim, and that's what I'm going to talk about mostly here, is to produce a female-specific attractant for an attract and kill system. This attract and kill system is not going to replace the sterile insect technique, but we think it could be an important part of an integrated pest management strategy. We would also like to get an attractant that would be more male specific, because, like I said, the males are the ones out there that really need to be traced in the landscape, because they're the ones that are doing the SIT work, and we'd like to reduce non-target catches. So with that, it's a pretty straightforward process, what we're doing right now. We need to identify putative attractants. So I should have pointed it out a little earlier, but when I was talking about swarm lure, there's 10 components in that swarm lure, 10 chemicals, and that has not really changed much over the last 50 years. So we really would like to identify novel chemicals and improve our attractants. So we're kind of starting from scratch. So this involves identifying novel chemicals. We use gas chromatography, mass spectrometry to do this, and we will analyze relevant host odors, skin odors, wound odors, other animal odors, feces, urine, because they could be cueing in on just the animal smells alone for long range, and when they get closer, they're cueing in on the wounds. We're also looking at beef liver. Like I said, that beef liver is interesting in that the captures, when we bait our traps with beef liver, they're females. We used beef liver when we were down in Nicaragua a couple years ago, and we did not catch a single male. Those 31 were only wild females. So we have a lot of, we'll call them ecologically relevant odors that we need to characterize. And the next process is find out if they respond to those electrophysiologically. Does it many of these chemicals they may not even be able to detect in the environment. So then we would test these in the laboratory and olfactometers, trap assays, and then once we have some candidate formulations, we would take those out into the field, which we do. Typically what we've done is we release sterile flies, so many sterile flies, and we recapture them to kind of assess some of these attractants or traps. I kind of like this. So this was from a paper where we tested swarm lure 4 versus swarm lure 5. But we wanted to know the number of non-target captures. Up here in the top, this is just, we did 3 releases and then we recapture them mostly within about the first 5 or 6 days after you release them. But we also monitored the Cochliomyia macellaria captures, which were quite high. And it was interesting. I just thought this was interesting that we, very locally where those traps were, we really did knock those populations down quite a bit over time. By the end of this was over a 26 days effort. And by the end of that time, we had knocked those populations down to where we weren't capturing anymore. So I think it's very important to understand how insects navigate in the environment. And for those of you that are not entomologists, the fly's nose is basically, if you can see my cursor here, it's the antenna of the insect. We call this the arista here, but this here is the third segment of the antenna. And if you zoom in, these are SEMs, scanning electron micrographs of the antenna, that third segment of the antenna. You can see various types of sensilla. This one here is what we call a coeloconic sensilla. This long one here is a trichoid. And then you've got, this is another type of sensillum here, and this is a basiconic. And you can see the pores where odorants actually enter. So again, this is another, this is actually on a palp of a mosquito, but what happens is chemicals go through these pores and they bind to receptors on the surface of these odorant olfactory receptor neurons. And then when they do that, they send a signal to the brain. And these odorant receptors largely, almost solely determine the specificity that of what they can smell. It doesn't mean it's going to determine necessarily how they respond to that smell, but based on the binding of that in these basically cation channels, this is how, and it's not too much different than the way we smell, right? We have olfactory or odorant receptors in our noses. They're not really related to insect odorant receptors, but they function in a similar way. Some people can smell things that other people can't smell. So it's the same concept. So I told you that the first work I did in New World Screwworm, I annotated the chemoreceptors in New World Screwworm, and then we also annotated them in Cochliomyia macellaria, and we can learn quite a bit from this. We have a few more in both the Cochliomyia species than we do in Drosophila, but the power of this is we know a lot about these Drosophila odorant receptors. Many of these have been well characterized. So many of these we kind of infer their function based on how they cluster with other odorant receptors of known function, or at least it gives us a hypothesis. And one example, and I'll talk about that in a few minutes, is it's hard to see, I realize that, but right here at the top of this gene tree is Drosophila OR67D. This is the pheromone receptor for cisplacenyl acetate, a pheromone in Drosophila. And we've got 3 closely related receptors there. So one of our hypotheses is that these are or could be pheromone receptors. As of now, actually over the course of 100 years of research, no one has identified a volatile pheromone in screwworm. We have evidence that they're there, but they have not been characterized. There's been some work on non-volatile pheromones, and that would be more through taste receptors, but this would be for volatiles. So they would be smelling these. So we can learn and make predictions based on how these are related to Drosophila odorant receptors. So keep that in mind as I go through here. As expected, hominivorax or New World screwworm had fewer odorant receptor genes than Macellaria, kind of going back to it doesn't do as many things as it's kind of look at hominivorax as more of a specialist and Macellaria as more of a generalist. And also, I put this in to demonstrate that even though we say there's 82 odorant receptors in New World screwworm, functional odorant receptors, or 85, it's really variable. This is work we did on sandflies, and these are just different populations. I had to kind of take a lot of this out because it's kind of a hard figure to see, but basically, this was on copy number. So this is an odorant, each line across here is an odorant receptor in, this is Cochliomyia longipalpus, and different populations of those in Brazil. And it was very surprising that what we found were these are each individuals in the columns, and the variation, even within the single population, was surprising that we saw. So when I say there's 85 odorant receptors in Cochliomyia hominivorax, that's what's in our reference genome. But in reality, you go out and start looking at each individual, you're going to see quite a bit of variation, is what we've learned. We did the intestinal transcriptome, so basically we want to see how these are expressed. We found the 80-some odorant receptors. We identified those in the genome, but which ones and how many of those are being expressed in the antenna? Which ones are the most important for the adult, right? Because the adult's stage generally is olfactory behavior is by the antenna. There are secondary olfactory organs, but largely by the antenna. So we did the intestinal transcriptome. We found 44 ORs that were at least moderately expressed above a given threshold. I think it was 5 CPM and above is what we considered, okay, it's expressed. You get a lot of these that just show very, very, very low expression. We don't know how relevant that is, biologically relevant. But one of the things that really was striking here is there were 7 odorant receptors here that showed virtually no expression in males. So this is very unusual. Other people have done intestinal transcriptomes of other blowflies or other houseflies, Drosophila, and we just never saw this before. So 7 of these are just in females. So we are in the process of leveraging this information to move forward to try to develop a female-specific attractant. So our hypothesis is whatever these receptors are tuned to, whatever they're responding to in the environment, whatever those chemicals are important for females and hopefully attractive to females, but not males. So this is taking an approach that we call reverse chemical ecology, where we can take those receptors and we identify something interesting in these odorant receptors, and then we kind of go backwards, right? A typical chemical ecology, you start with chemicals in nature, and you work in one direction to try to get to your end goal. In this case, they call it reverse chemical ecology because we're kind of moving in a different direction. We're starting with the odorant receptor. So how do we do that? And here's I want to mention this before I go too far. Since we also did Macellaria, the secondary screwworm fly, we did the intestinal transcriptome there and saw major differences. So it's even different in Macellaria, closely related fly. Some of these, like OR62, similar kind of a profile where males express this a little bit in their antennae, but most of these are just vastly different in their expression levels. Only two of the Macellaria were not or highly female biased. Now is OR7 and OR62. We also saw some other major differences between Macellaria and hominivorax, or secondary screwworm and New World screwworm. This is also very interesting to us. The most highly expressed odorant receptor in Cochliomyia macellaria is not even expressed in New World screwworm. It's a pseudogene. So this is very interesting to us. Perhaps we could identify what this responds to and make sure that it's not in our attractant, perhaps to make it more specific to New World screwworm. So we're various strategies for identifying novel chemicals. And another here is the one I pointed out, the potential or putative pheromone receptors in hominivorax. Like I said, we have not identified any volatile pheromones at this time, but we believe they exist. And we're working to identify them. It's kind of interesting here. You'll see there's three New World screwworm receptors that are closely related to the Drosophila pheromone receptor, but the other three species don't have this OR65, and they have a different receptor. So this is very interesting to us too. So perhaps this was part of the evolutionary process that kind of helped split them apart in nature during speciation. So how can we use these data to move forward with our studies and identify pheromones, identify novel attractants? So they call these systems heterologous expression systems. The one that I have been trained in and I am currently using is the empty neuron. So in Drosophila, they had identified natural mutants that they had basically that did not have odorant receptor OR22A, which is, you can kind of see here, this OR22A. We know which syncyllum it is. It's in Drosophila. We know what the other odorant receptor is in Drosophila. And we are now able to take these odorant receptors from New World screwworm and clone those into Drosophila. So now we can express those others that I showed you, those putative pheromone receptors, those female biased receptors, there's male biased receptors. So we can clone those into Drosophila empty neuron, and now we can go to those receptors or those syncylla in Drosophila and start testing different chemicals and to find out what chemicals are those odorant receptors tuned to. So very powerful. There are other systems, other heterologous systems such as oocytes and hex cells, but this is what we chose to use. This is kind of what the rig looks like that I do my recordings on. This is how we mount that fly, the Drosophila fly. So this has, we're going to say, odorant receptor OR34. And this is what the Drosophila antenna looks like. And we take a recording electrode. And it's these little finger-sized projections here. These are the large basic conics where those are expressed. And then we're able to just run panels of chemicals through there to see, hey, if they respond to this blend, this alcohol blend, then we go back with each alcohol that's in that blend to try to identify which alcohol that it is tuned to. That kind of helps expedite this. And we also have the ability to do odor extraction. So here's an example of a flower that is as you can see, these flies are very much attracted to this flower. So we can find chemicals that are in these flowers. We can find chemicals that are in this beef liver or other flies, and we can start analyzing those and putting those across the antennae. So this is where we are right now. We are screening these odorant receptors with a series of different animal odors, fly odors, flower odors. We're going to start working on wound odors as well. And this is some preliminary results. So we're not through with all of our screening, but this is OR34. You can see it's the female biased, actually upregulated in gravid females as well. And what's given us some optimism here is, so here's, if we puff just beef liver, we get a nice response. So we're looking at this depolarization, this distance from, if you can see my cursor, from this, where we hit the chemical, that it's a flat line, then it, the depolarization goes down. That's kind of the amplitude of our response, 13.9 millivolts. And if we do swarm lure 4, we don't get much of a response, very little. So this is telling me there's something in beef liver that this odorant receptor is tuned to, or it excites this odorant receptor. And it doesn't look like, it could be one of the, it could be something in swarm lure, but it does not look like it's really, swarm lure is doing much excitation. And then we found an alcohol there that actually gave even a larger response in beef liver. So this is the approach. I think it's panning out. We have to do all the behavior, but right now we're screening odorant receptors, screwworm odorant receptors for novel chemicals, and then we start testing those in the laboratory down in Panama. So we're very excited about this. Some of the things in the future is we can take these extracts, these odor extracts, and we can run those through. Here you can see my recording rig. This is a GC with FID. So we can inject some of these odor extracts, because there are a mixture of various chemicals in these extracts. These odors are not typically one single chemical. But if we can link this GC with the electrophysiology and go through, and here's an example of what we can do. So here's the antennal response at the top, and at the bottom is the chemical composition. So in this case, you'll see this is a response to this chemical right here, which that quickly allows us to eliminate these others. Normally, I would say, oh, I would test this here, I would test this. This would be the first thing I would try to test. Well, it didn't even respond to that. And again, another advantage of the system is here's 2 smaller responses, but it must be highly sensitive to these because they don't even show up. They're not obvious on our chemical profile. So there's a lot of power in this. There's also challenges here, but this is where we are going to next. We have the system set, purchased and acquired. We're just got to finish setting it up, and we will start doing more of this, what we call GC linked single syncytium recordings. And I believe that's pretty much everything I have. this has involved many people, from genomics to um, chemists. Just many people have contributed to this work. So um, I think that's it. If I'd be willing to take some questions now, if anybody has any questions.
Fantastic. Thank you very much for the talk. That was great. And I just want to remind everybody, if you have questions, go ahead and throw them in the Q&A section, and we'll go ahead and address those. I see we have two now, but I had a few questions watching your talk. Um, so, and maybe you kind of covered it and I missed it, but so it makes sense to go to, picking up on chemicals from the beef liver or plants they might interact with. Do you screen any synthetic or artificial chemicals along with that?
Yeah.
Yeah.
So yes, if I go back here, so what we've done is we've assembled a panel of chemicals that based on animal odors, based on decomposition. So we have assembled about 70 into this panel, right? So that's what this is here. Um, but there's just so many chemicals in these in nature that what we're finding in some of these receptors is we'll screen all 70 of these chemicals and we don't get a response. But we take an extract and we get a large response. So we got to kind of tease that apart a little bit. We believe in many cases they're probably responding to more than one thing in that blend, but we should see if they respond to any of these chemicals, we should see that in our electrophysiology. So it's kind of interesting, just amazing how many chemicals there are in our environment, what we smell.
Right. It is kind of wild. And I imagine you'd probably also see some synergistic effects with multiple compounds as well.
Absolutely, yeah, yeah. We expect to see quite a bit of that.
Right. And just a follow-up question on that. I'm fascinated by AI, so I always ask it an AI question. Have you guys thought about using kind of AI to reconstruct the protein folding of the olfactor receptors, or have you thought about that at all, or is that a tool you're interested in?
Yeah, I mean, I get asked that quite a bit.
Sure.
I don't know. So I think what it could do. So in the case of this receptor right here, OR34, we were kind of these results kind of match what they found in the closest receptor in Drosophila. So there are some things that we can take home from that, right? But so far it's mostly been phylogenetic relationships, right? How closely are those receptors related? Some of these they have a one-to-one relationship. They're conserved through many of the Diptera, and they respond to the same chemical in all of the Diptera, right? So that ability is out there, but to model it without having that baseline data, I think would be challenging. But I think as we collect more data, we can say this is what this structure does. Help predict what this structure does, right? So we kind of need to build a foundation. But some of these we probably already have a good idea. The endollergic receptors, very highly conserved in the flies, mosquitoes and blowflies and Drosophila. Um, we none of these are on my list. We will probably eventually do them to confirm that's what they're doing. But like I said, I'm looking for things that are more novel, unknown. Because I think we're just with those 10 chemicals in swarm lure right now, we're missing. There's a lot of area for improvement, I think.
Oh, there you go. Okay. Here's our first question from the audience. Maybe maybe a little bit of a trick question. The question is can you tell us a little bit about the feasibility of female only SIT? Now with SIT you're releasing sterilized males.
Right, right. Yeah. So females, ideally it would be great to just release males. That's very challenging. You could sort them at some point. Um, we're talking 100 million a week. We've looked at ways to sort just by sex and just release males. It's just so impractical at that large volume. And most of the sorting systems that like the mosquito people are using, they're sorting very early. I believe they're probably sorting at different stages, but I think ideally you would want to sort at the egg stage or very early, late embryo stage,
Sure.
um, because you don't have to feed those any longer, right? So
Right.
sorting at the adult, we have 100 million flies. We're going to just sort and throw out 50 million, right? But it would be great if you could sort at in the embryo and say, hey, we just chose 100 million males. We're not quite there yet. We'd love to be, but yeah, it's a taller order. A lot of people think that shouldn't be that hard to do. Yeah, it's at 100 million a week. Sure, you can do it with maybe if you label males somehow on the Y chromosome. And that way you can pull them out with some type of a sorter. But to do 100 million of them is going to be a challenge.
That's a tremendous number, and I'm not sure exactly what it's like in New World Screwworm. I remember sorting 80s Aegypti, and it's like thankfully the female pupa are much larger, so it's
Right.
easier to
Yeah.
sort them.
Yeah.
Um, but maybe hopefully eventually we'll get some sort of fluorescent marker that allows us to distinguish the males from the females, or something
Right.
along those lines.
Right. Right.
Here's the next question: is there a way to take the captured females that are on the aged beef liver trap and determine which receptors are overexpressed to find female-specific odorants from the beef liver?
Yeah, and that's a good question. I mean, that's something that we've talked about. I don't think the question is, are they upregulating those receptors in the presence of beef liver? And I think those ones that are highly expressed on the antenna, they're probably just expressed in a lot of sensilla on the antenna, right? So I don't think it's their, but people, we have talked about this, right? Does that expression change in the presence of whatever the host odor or the beef liver? You kind of would think of it as they're already got their repertoire of odorant receptors expressed, and they're basically, we feel like the highly expressed ones. The reason we're finding a lot of expression is because they're just in a lot more sensillas than the other ones. Yeah, I don't, but we have talked about that. Actually, talked about that pretty recently. So good question. We don't know. I mean, if it's going to help us, but we probably will do something like that where we express one thing to beef liver and another thing to something else and dissect their antennae and sequence them.
Very cool. And I guess I have a follow-up question based off that, just thinking about the question, and I'm not sure, right? I'm not an olfactory expert, but and I guess if expression levels are pretty static, then it wouldn't change. But I wonder if a female went longer without getting that protein meal and she kind of became more desperate to
Right.
find it, would you see higher expression at that point?
Yeah,
So
that's
if
another
you
good question.
starve them.
Yeah, that's another good question. So the reason we chose those days that we chose to do our gene expression analysis were days 3, which they're mating heavily right then, right? They're full on mating on day 3. So we're interested in kind of what their gene expression looks like in males and females at the same age in different states. As you saw, we did mated females and unmated females on day 3. We didn't see a big difference there. So we didn't see a big shift after, but maybe that does occur a little later on. What if she has not been mated? Does she start changing her expression profile, right? So kind of along the same lines.
Right.
Um, I would think it would even be more so with, we also did that 6-day post-adult eclosion in the females that have been mated, and they should be ready to go lay eggs. They're ready to lay eggs on day 7. So we were curious do they change their expression at that point? I think we will eventually do change things over time, or analyze things over a more fine time point series, right? Day 2, day 4, day 6, day 8, and just that would be a neat approach to see if they change anything in particular, if the females haven't been mated yet or haven't laid eggs. We feel like hypothesize that sometimes females will lay eggs in not the greatest places, right? But I think they get desperate once they have to
Sure.
get those eggs laid. I think mosquitoes do the same thing, that
Yep.
they may lay eggs where they normally wouldn't lay them, because they know the mother has offspring, to lay. She's going to put them somewhere before she loses them. So, yeah, so that's another thing that we're interested in. Um, yeah, but
Cool.
yeah, good questions on. We will. I'm sure we will do a time series at some point. We'll also look at other tissues as well. The maxillary palps, mouth parts, ovipositors. We
Right.
plan on doing that this year as well.
Awesome. Here is the next question. Thank you for this very interesting talk. This may be a crazy idea, but could RNAi-based biopesticides, such as Leprona, developed against the Colorado potato beetle, be developed against female-specific odorant receptors, such that these receptors are knocked down and the females find it very difficult to find their target animal? Your thoughts, please. Thank you.
Oh, so this would be on. So this would be on the released females, so they don't.
I guess you could use
Never
it for
never
released
even thought about it.
females, or I guess if you had an area-wide spray that could kind of inhibit their ability to find
Right,
hosts.
right. I mean, the most logic or the easiest way to do that is do the genetic manipulation on the strain, on our mass produced strain, right? And then make it to where, yeah, they don't go near the male. Because if you could keep them away from the males, right? Um, yeah, that's a good question. And getting it into the wild, yeah, so these guys are really difficult to target in the wild. They don't spend much time on animals, right? Because they're not blood feeders.
Sure.
The only time that they really would visit an animal is one, if the female needs to lay eggs, she'll lay them on an animal. But they don't go there to take a blood meal. If they've got a wound, they might go there to lap up some protein, from some blood source or a wound. But I think and I'm sure that happens some, but males won't be on the animal at all. It would be rare, and females would be rarely on the animal unless they laid eggs. So it's hard to get at those adult females if you want to kill them. Um, and they are susceptible to the traps and lures. So that's just how we've done it. Just broadly spraying on animals, probably not very effective, or in vegetation, right? It just doesn't, and then they're not in high density either. So they're a challenge to get at. They are very challenging. I know people are looking into other methods to control screwworm, but they are a challenge because it's not like a mosquito where they're going to be on people or animals for a blood meal. They just don't do it. So
Right.
yeah.
And that I kind of had a question similar to that, and you kind of touched on it a little bit, but maybe you can expand. I guess, coming from a lab where we knocked out genes and modified genes all the time, do you have any thoughts on how these olfactory receptor genes could be modified or overexpressed, knocked out to be used? And I think you kind of hit on it a little bit. It would be interesting, right? If you had a strain and you could target female specific receptors, maybe you could, if you're gonna eventually release females anyways, because sorting's such a challenge, you could have a female strain that's less likely to find a food source.
Yeah, survive. Yeah. So we have done some functional characterization. I didn't do it. Max Scott and Danny Paulo knocked out Orco, which is the odorant receptor co-receptor, and this is some of the evidence that we see, or some of the evidence we think suggests there is a pheromone. Is when Danny knocked Orco out, they didn't mate well. They didn't do anything well, right? So. We will probably do additional functional characterization by knocking some genes out. Um, Perhaps down the road, you could do some knock-ins to test variants, right?
Mhm.
Um, but right now, so interestingly, Max Scott lab knocked out one of our female biased receptors, but in Lucilia caprina, and it had a large effect on the fly's ability to do anything. So I think it's a powerful technique. We It's harder to kind of transition that into a mass production line and
Sure.
releases because of there's a lot of approval processes. Um, but definitely for just some questions, right? Just to answer some questions about, odor receptor variability. I think we will do more of that for sure. Yeah.
Very cool. All right. Here's our next question. Not sure if you mentioned it, but did you look at wound volatile profiles from different types of wounds? Are you testing chemosensory attraction of gravid females or females in general? And I think you had a little bit on that in your slides, but if you could
Yeah.
expand.
Yeah. So we do, when we test in the lab, we test on mated females, unmated females, and then we test on what presumed gravid females, right? They've been mated and they're about on day 7. Um, so we do test on those. As you noticed, there were a lot of changes in expression associated with the females being gravid. I think the next question is um, first characterizing those receptors, but maybe look at, are these just changes associated with they're getting older? Maybe this is not anything to do with oviposition, but maybe would a female still, would we see that expression pattern in a female that's not mated and not ready to lay eggs? So we'll look at things like that, but we're very interested in the gravid females. Um, and that's something that we've never really been able to target up to this point because they don't come in to lay eggs on beef. So they will come and take a protein source. So they've probably been mated. So they technically are gravid at some point, but they're not really ready to go lay eggs. So um, definitely would like to get those gravid females out of the population because they're going to be wild females. If they're gravid, they're wild females typically. Um, Definitely, yeah, for sure. We are very interested in the gravid females.
Our next one is actually a clarification, which is I didn't know this, and I've learned something new today. And the comment is remember that male only SIT is not universally the best. Pink bollworm was eradicated with a mixed sex sterile population. The sterile females actively contributed to pheromone mating disruption. So mixed sex
Um,
SIT is species specific as to whether it's helpful, harmful, or neutral. So I am
yeah,
biased because I came from a mosquito lab. You don't
yeah,
want to release any females.
and that is definitely true, right?
Yeah,
I it's a largely different, right? They're very pheromone driven when you look at the bullworms. We don't know how much that's going to change, right? So it's just been estimated or predicted that a male only system would be superior, right? Then it may be just slightly superior, but there's always a chance that it may not be. It may be slightly inferior or major. Yeah, I mean, I get that, but their pheromone systems are quite a bit different.
sure.
If if we work it out here, I don't think it's going to be anything that's long range. Typically, these fly pheromones aren't long-range pheromones like you'll see in other insects and beetles, bugs, Lepidoptera. But it could be, right? He's right.
Right.
It could be that all males may not work. But we do know that if we can get females, those wild females out of the population, we're pretty confident that that would, it definitely wouldn't hurt. I think anytime we can get those gravids out, and also remember these are in pretty low density. So you may think, well, you only caught 100 flies. Well, maybe that's all that was there locally, right? So when we did our trapping down in Nicaragua, we were trapping near recent cases, and only two sites produced screwworm, and we were trapping in only one site. So it's also very different because of the low density in nature too. So that also
Right.
begs the question: how are they finding each other? I mean, that's one of the things that we're interested in. There's very low density in nature. How are males and females finding each other, right? So there's, I suspect there's some kind of a marker system, whether it's an olfactory mark, like Culex, it's a visual mark, right? And they swarm. Flies are just, they're different. So it could very well be that it's, Aedes aegypti, I believe the system is. The males are still attracted to hosts because that's where they find the females. I think that's one of the mating systems for Aedes
Oh, yeah.
aegypti that they described. So there's another hypothesis that says, well, maybe males are still attracted to animals because that's where they're going to find females. The system's not, it needs a little working out. All good questions though. Those are the questions we're asking right now.
You also in mosquitoes, you see wing beat frequency is an important thing.
We've looked at that too. So
Mhm.
we didn't find much variation between strains and between males and females. And I had to go back. There's going to be a paper come out on that actually pretty soon.
Very cool.
They did that work down there in Panama at Copeg at our production facility. I'd have to go back and review it. But yeah, we were very interested because one of the some people say they believe the male is waiting near whatever this marker is, or what they're waiting on the female, and some people say they will fly up and clasp the female as she flies in, so that he would have to be able to recognize that that's a female screwworm, right?
Mhm.
But we're not sure how. It's hard to many. Apparently, this I've never seen them mating in the wild, but I haven't spent a ton of time in the field. But it's pretty rare for people to see them mating in the wild. Very few cases have been described in the literature, but just a big gap.
Interesting. Let's see. We have one more question and maybe a follow-up from me. But this is an interesting one, and I think it hits on an important topic. It's a bit eccentric, but could tiny cameras be attached to these little critters to find out where they hang out? That might also provide some insight on mating.
Yeah. So maybe not. I don't know about cameras,
Mhm.
transponders. We're looking into that as well. There's a research group that does that in taphrydids, in the fruit flies,
Mhm.
and we are kind of in talks with them about, because then you could tell where they're hanging out in the environment, right? Because we know when we trap them, they're usually near water, they're usually in heavy vegetation. But I think that would be, we are interested in where they are. Like I said, they're hard to find. They're hard to get and predict where they're going to be in nature. But yeah, cameras would be even better. But yeah, I don't know. I don't know that,
Maybe
but I think it would be great.
I
Um,
I think they used transponders to track down the giant Asian hornet when
yeah,
it
they may
was up
have.
there in Washington.
Yeah,
Yeah.
yeah. But you could ask some really good basic biological questions with a transponder. Um, just put them in large enclosures and track how they're moving. If you get a male and female, or a group of flies. But yeah, so that's something that we're also interested in.
Very
But
cool.
there are
And
little
I,
dinky transponders. Yeah.
I think my follow-up question on that, because I find, right, I'm a geneticist by training, so often I'm leaning in towards the how could you genetically modify it, all that kind of stuff. But I think, I like the AI questions. I also like kind of robotics questions because I think really there's a lot going on in AI and robotics, and I think like traps are really important, especially right now as we're trying to monitor where they are and prevent them from crossing the border. But have you thought of any way of integrating robotics into surveillance or traps or anything along those lines?
Um, I know people are interested in that, right? So it would be, what's the term? A biological surveillance,
Mm,
where it's kind of like a smart trap, right?
right.
So, because if it's for surveillance, all we need to know is if we have screwworm in the vicinity. The challenge again becomes, how do you get them into the trap, right?
Okay.
So that's why my focus is on getting into the trap. I think that opens up some doors for us down the road. But yeah, I know people are considering various strategies on, can you do surveillance? It would be remote, right? You wouldn't be checking the trap every day. Yet if this fly, if a hominivorax flew through or flew to this trap and it identified it as that, it would transmit that information via some other system, right? Electronically. That would be awesome. But the problem again is getting them to the trap.
Yeah.
So yeah, I think if we can improve our attractants, it's going to open up some more doors for us.
Oh, for sure.
Yeah.
Because then you just cover such a larger area. Yeah, it's
Yeah, yeah. I
very
mean,
important.
surveillance,
Hmm.
mating, various aspects. So that's where my focus has been, just better understanding the olfaction. And I think those other things will come, but I just feel like I it's going to be hard sell right now when we can't really get them. They don't can't dependably get them to the trap, especially a male. Um, but yeah, I think those things will come.
Getting a better attractant is the first step.
Yeah, and I mean, yeah, in my opinion, because that's what I do. So, to be able to move on with some of these other questions, right?
Right,
I mean, I
right.
think that would be very powerful. But if you can't get them there, how are you going to get them photographed or get them phenotyped or get them whatever, right?
Right.
It's going to be hard if you can't get them to your right.
To your
Um,
trap.
so yeah, so I just think these will be more feasible as attractants, but I'm sure people are working on it, right? But I think that will get more powerful as the attractant gets better,
Right.
right? Yeah.
Very cool. All right. Well, I think we've hit all the questions in the Q&A, so I'll go ahead, and we're almost at time. I'll go ahead and wrap us up. So, thank you, Dr. Hickner, for an excellent presentation, and thank you to everyone who joined us today and contributed to the discussion. This webinar is part of our ongoing series, The New World Screwworm: Past Lessons, Present Programs, and Future Frontiers, and we'll hope you will continue to join us for upcoming sessions as we explore additional perspectives. Please stay tuned for announcements regarding next week's session. So if you've looked at the sign up, next week is TBD. We're still not 100% sure yet if it will be going on, but updates will be shared by the Gene Convene Virtual Institute newsletter. So if you're subscribed there, you'll get an update. Any of our LinkedIn pages we'll share there, or through Zoom registration communication. So if you're registered for it, we'll send an update if there is a cancellation. If there's no update, then we'll be going ahead as scheduled next Wednesday. So we encourage you to follow these channels just to ensure that you receive the most up-to-date information. Additionally, we invite you to visit the Gene Convene YouTube channel, where the first two webinars are now live, and the Gene Convene Virtual Institute if you are interested in learning more about genetic biocontrol. Also, please feel free to subscribe to our newsletter to stay informed about future events and activities. Thank you again for your time and participation, and we look forward to seeing you at the next webinar. Have a great day.